So today I'll be talking about my gut microbiome project that me and my partners have conducted over the past few weeks. We have looked at the different types of microorganisms that are present in the gut microbiome and the proportions of those microorganisms. It is important because the microbiome shows a lot of correlation to health issues. It shows correlation to the animal's environment, the food that they eat, the microorganisms that are present within their bodies. And this shows a lot of correlation to a lot of diseases that humans are going through nowadays, such as IBS, which is irritable bowel syndrome, which is characterized by chronic abdominal pain and discomfort associated with alterations of bowel movements in the absence of demonstrable pathology. When comparing an IBS patient to a healthy patient, an IBS patient will have a decreased amount of lactobacillia and bifidobacteria present. Will have an increased amount in aeros and musculobacteria will be more abundant. Also, when looking at obesity, which is characterized by proportions in the composition of function of the gut microbiome in humans, which is found in the small intestine. In a study comparing mice with obesity and healthy mice, it has been found that genetically obese mice have a higher proportion of intestinal firmicutes and half the amount of normal Bacteroidetes, and generally have a harder time degrading polysaccharide compared to the healthy mice. Both of these sources I have found on the World Journal of Gastroenterology. In our experiment, we looked at microorganisms present in two different huskies that differ in age. Husky 343 is 3 months old on a grain-free diet, while Husky 344 is 2 years old and 5 months while on a raw diet. We looked to answer what was the proportions of microorganisms present on both huskies. We hypothesized that the older husky, who was 2.5 years older, would be more inclined to have health issues regarding the gut microbiome due to its larger diet. When looking at what we did in the protocol of this experiment, we first isolated the DNA from the fecal sample using a Kagan kit. We then concentrated that DNA after purification using a Zemo kit. We quantified the DNA of those samples using a nanodrop UV spectrophotometer. Then using amplicon PCR, we amplified the 16S RNA gene of DNA by using PCR. Next, we used PCR cleanup. This procedure was conducted to remove PCR primers, which would interfere with the indexing PCR reaction. We will then index that PCR, which is conducted in order to tag and specify exactly the sequences in order to know which sample came from which when they're all mixed together. Then we did another PCR cleanup using Agincourt Ampere XP kit as we did previously. We quantified that DNA using indexing PCR products, and then we finally used DNA sequencing using a next-gen sequencer to determine the sequences of our DNA samples. We also used gel electrophoresis, which separates charged macromolecules based on their size. This step was actually right after amplicon PCR and before PCR cleanup. I'm sorry for the mix-up there. When looking at our results, looking at the ratios of protein to DNA, our ratios pretty much stayed in the area of 1.6 to 1.8, while our negative control had a value of 1.2. The concentration of these samples seemed to be in a very narrow range of 46 to 50, while sample 1 had an outlier of a concentration of 1.03. As you see in our gel electrophoresis screenshot, that the DNA that we will be looking at had a base pairs of around 400 and 500, while our negative control did not show up, but the rest of them did. Looking at the different type of animals present in the bacteria in the gut of the animal 343, which is the younger husky, and then the animal 344, which is the older husky, we looked at, there was a lot of different amounts of reads. In the younger animal, their total amount of reads was measured at 3.023, while in the older husky it was measured at 9.234. Based on the older husky, there's a lot more bacteria and microorganisms present in every single column, from kingdom, phylum, class, order, family, genus, and species, compared to the younger husky. The taxonomic level value for each of these levels was around 1,500 for the younger husky, while for the older husky, the taxonomic level was around 7,000. And that is a huge difference. The classification for other and unclassified microorganisms that are present in the gut of these animals that we have no record of, this implies that there are many different types of microorganisms that play a role not only in our gut but in our entire atomic organismal lives. Lastly, when looking at our discussion, well, first we isolated the DNA using a Kagan kit. We concentrated that DNA using a Zemo kit. The concentration of our DNA is high enough. That means the purity of the DNA is higher. Protein absorbs light at 280 nanometers, while DNA absorbs light at 260 nanometers. Pure DNA has a ratio of approximately 1.5 to 2.2, while ratio below 1.5 indicates possible contamination. As you can see in our A260, A280 ratios, all of our samples conclude that that is pure DNA outside of our negative control, which is at 1.2, which means that there could be possible contamination. Out of the 6 products tested for a band, 2 did not. The positive control formed a band as expected, along with samples 885 and 711. This indicates that our PCR amplification worked, or gel electrophoresis, due to the base pairs and these markages, our DNA. Amplicon worked. The cleanup that we use will remove components of the Amplicon PCR buffer. It will remove unused primers. We added a unique set of short sequences tags to the ends of our amplicons from each sample via indexing PCR reaction. We sequenced that DNA so we will know which sequence came from which samples via those tags. We used a fluorometric assay to determine the concentration of our amplified sample after the PCR cleanup. These are way more accurate than the spectrophotomic assays. They are based on the fluorescence properties of a molecule after binding to the target, which they bind it during electrophoresis. So using the fluorometric assays largely based off of the gel electrophoresis that we did prior. There was, in our animal 343, it came out that there is 88.42% bacteria with 11.52% unclassified. Animal 343 had the species level at 65.38% unclassified. It had some Ruminoeuchinus navis, Asteria alberti, amongst other things. Animal 344 had a level of 86.27% bacteria with 13.7% of it being unclassified. Our hypothesis cannot fully be considered supported due to the fact that, assuming that a large lifespan directly correlates to the types of gut microbiomes present, that is not true. Other factors such as environment may affect the different types of microorganisms present. And here are our references. And that is our project. Thank you.